Microscopical techniques in microbiology. We'll start with staining. So staining is of primary importance for the recognition of bacteria. Information provided by the microscopic techniques will often provide rapid result to diagnosis of an infection. This is a clear protoplasm which is so feebly refracted that it's difficult to see in an unstained condition. Unlike dark-ground illumination is required. Alternatively, use of phase contrast techniques. Different techniques include wet mount preparation, gram stain, and zinc stain are done. First is wet mount and hanging drop technique. The hanging drop and wet mount techniques allow for the observation of living organisms. Examination of wet mount preparation is mainly used for detection of motility, capsule presence, and the vaginal yeast cells. These techniques are usually performed without the addition of any stains, therefore these organisms can be difficult to see. If you use these techniques to observe motility, be sure that you can tell the difference between motile and Brownian movement. Vibrations of the cell caused by the cells colliding with water molecules also should be identified. True motility always allows the cell to move in a different direction and across large area. Now, type of stains. First one is simple stain, which provides a quick and easy way to determine the shape, size, and arrangement of the cell. Here, crystal violet, methylene blue, or saffron stains can be used. Simple stains employ only one type of stain. Second is differential stain, which differentiates between two types of organisms. For example, gram stain, which differentiates gram positive, gram negative, and ZN stain differentiates between acid fast and non-acid fast bacteria. Next are special stains used to identify or stain special structures in an organism, for example, capsule or endospore stain. How to prepare a smear? So smears are usually made up from lesions, exudates, blood, tissues, and colony suspensions on an isolated bacteria. Smears to be stained are firmly fixed on the glass slide before various dye solutions are applied. Slides must be cleaned by soaking in 95% ethyl alcohol, then wiped dry using a clean glass, otherwise the material, that is the smear material, may wash off. Make a direct smear or film on a clinical material. The material should be a thin one. Bacteria from solid media are transferred to a drop of distilled water or saline on a slide. A loop full of broth culture is directly placed on the slide. Allow the film to dry in air. Drying is accelerated by using an incubator. Fix the film quickly by passing the slide through the Bunsen burner several times. Proper fixing also prevents washing off. So this is how the smear is prepared. I think you can all see this. First one is gram stain. So gram stain is one of the most important staining procedure in microbiology. It is used to identify between gram positive organisms and gram negative organisms. Hence it is a differential stain. I think everyone knows this. Gram stain involves four parts. The first one is crystal violet, the primary stain, the gram iodine, the moderate decolorizer made up of acetone and alcohol, and saffronin, which is a counter stain. So step 1. Here there is application of the crystal violet on the prepared slide. So after around 1 minute, add iodine, which is grams iodine. The function of grams iodine is as a mordant. Wait for 1 minute, then add acid or alcohol, else acetone can also be used for around 2 to 3 seconds. Wash it off, then apply saffronin as a counter stain and wait for 30 seconds and wash it off. So every step starting from 1 to 4, the slide should be washed off after every stain is applied. I think it is clear. Good. So the protocol. So prepare a bacterial smear. Saturate the smear with crystal violet for 1 minute. Rinse the slide gently. Saturate the slide with iodine for 1 minute. Then wash it off with water. Decolorize with the gram decolorizer. Either it can be acetone alcohol for around 3 to 5 minutes. Acetone can also be used. Okay. So only if you allow the decolorizer too long, it will be bleached the crystal violet out of a gram positive cell. So care is required. Then rinse the slide with water. Counter stain with saffron for 1 minute. I told 30 seconds. 30 seconds is more than enough. Rinse the slide gently with water. Carefully blot the slide dry with biblious paper. Observe the slide under the microscope. Use proper microscopic techniques with special and proper staining care. You can see that the gram-positive bacteria will be purple and gram-negative will be red or pink in color. So what are the differences between gram-positive and gram-negative cell walls? Gram-negative and gram-positive cell walls are distinguished from each other by the differences in the cell wall. The differences affect the way the cell takes up and retains the stain. The cell wall of the gram-positive organism includes a thick layer of protein sugar complex called the peptidoglycan. This is around 60 to 90% of the gram-positive cell. Peptidoglycans are present in cell wall of gram-negative, but they are only 10 to 20%. Gram-positive cell take up the crystal violet, which is then fixed to the cell with the help of iodine mordant. This forms a crystal violet iodine complex, which remains in the cell even after decolorization. Decolorization of the cell causes the thick cell wall to dehydrate and shrink, which closes the pores of the cell wall and prevents the stain from exiting the cell. At the end of the gram stain procedure, gram-positive cells will be stained purple. So this is how the gram-positive will look. The first two images show the gram-positive bacteria will look. The first is a cocci, the second was a bacilli. So the purple color means gram-positivity. The second row, okay, the third and the fourth smears show that the pink color slides. These are gram-negative bacteria. The first one was a cocci and second was a bacilli. So this is a better picture where you can see gram-positive cocci in clusters are seen. Example is Staphylococcus aureus. Understood? And this is gram-negative bacilli, which is pink in color. Take note of everything. And this one is a gram-negative cocci. You can see that it is present within the cells. So this is intracellular gram-negative cocci. For example, most common is gonococci. Very dangerous. And this is a gram-positive diplococci. Diplococci means two cocci seen in pairs. Example, pneumococci. These are some other pictures. Next one is Zielinson's method. So this is also called as acid-fast method. This is hot stain method. Here the acid-fast stain is used to differentiate between the acid-fast organisms such as the members of the genus Mycobacterium. Acid-fast organisms are characterized by a wax-like, nearly impermeable cell wall. They contain mycolic acid and a large amount of fatty acid, waxes and complex lipids. Acid-fast organisms are highly resistant to disinfectants and dry conditions. Because of the cell wall is so resistant to most of the compounds, acid-fast organisms require special staining techniques. The principle of staining depends upon the resistance of this organism to decolorization by alcohol, acid, because the cell wall may contain waxy material, that is mycolic acid, which prevents the removal of the carbon fusion from the cell wall. So, as prior, this is how the smear is prepared. So this is step 1. You cover the smear with the carbon fusion and give intermittent heat for around 8 minutes. After the slide has cooled, decolorize with acid alcohol for around 15 to 20 seconds until you can see that at the beginning of decolorization the solution is yellow in color. You should decolorize until it becomes transparent or pure white. Okay, so the third step is stop decolorization action of acid rinsing briefly with water, then counterstain with methylene blue for 30 seconds, then wash it with water and blot dry and examine under a oil immersion microscope. Oil immersion microscope is 100x microscope. Fine. So the protocol. The primary stain used here is carbon fusion, which is a liquid solid and phenol containing component which can penetrate the cell wall. This is further assisted by addition of heat. The smear here is rinsed with very strong decolorizer which strips the stain from non-acid fast bacteria but does not penetrate the cell wall of the acid fast organisms. The decolorizer is a non-acid fast cells can take up the counterstain methylene blue or other stain which can be used here as counterstain is malachite green. So the steps: flood the slide with the strong carbon fusion and heat until the steam rises but do not boil. After 3 to 4 minutes, apply more heat until the steam rises. Do not let the stain dry. About 5 to 7 minutes of intermittent heating is required. Decolorize the acid alcohol until traces of red have disappeared. As I told you, it should be pure, colorless. Okay, so decolorization should not be attempted in one stage. There should be intermittent washing with water and reapplication of acid alcohol. Wash with water and add counter stain with malachite blue. Sorry, students, methylene blue for 1 minute. Wash and stand M to drain. So the acid fast organisms will be pink in color and the other organisms will be blue. So it can also be green when malachite green is used. Fine. So the pink color beaded organisms are acid fast bacteria. When you can see that the malachite green stain when used, the background is